right, so thank you very much, Anna, and thanks to IDG for supporting this work. So to go along with the theme, so I made a bigger title saying Illuminating Chloride Intracellular Channels, but I do promise that I'm not going to talk about every single channel. If you remember from my in-person meeting, I talked about CLIC6, which is one of the family members. Today, I'm going to mostly focus on a chloride channel, which we just published in mitochondrial associated membranes, and this is CLIC4. So let's talk about it. So all the work I'm going to show today is done by my very talented postdoc, Dr. Devasena Ponalagu, who will be starting her lab next week in University of Washington, Seattle. So there are 2 positions open, one in my lab, one other lab, in her lab. So if you are interested in chloride channels, please contact us. All right. So most of the work on ion channels, as you all know, are, well, published even today. Most of it is on these channels sitting in plasma membrane. What I always try to tell my students and everyone, trying to convince everyone, is that almost 95% of the membranes are sitting inside the cell. They are on these organelles like Golgi, ER, mitochondria, nucleus, and so on. So there are channels present there. And believe me, the membrane potential of mitochondria is quite negative. It's negative 180 millivolt almost. And here it's negative 60 millivolts. So to maintain that sort of membrane potential in these organelles, you have to have channels and transporters. And they are understudied, most of these channels. And I'm going to touch upon anion channels today and specifically CLICs. And as you can see in this figure, in mitochondria, our work and others, other groups have published that there are several of these channels sitting there, like BK, we published that, Brian O'Rourke has published, and other groups have also published. Then MCU, we are all very well versed. But if you go towards anion channels, the only anion channel well characterized in the outer membrane of mitochondria is VDAC, and the inner membrane is IMAC. We don't really know the identity of this channel yet. And in the associated membranes, we know there are lots of calcium channels like ryanodine receptors, but again, there are no chloride channels very well characterized there. So it's not like they don't exist. These channels do exist. The only issue is we have not studied them yet, or we have not characterized them. So our lab is focused on trying to figure out what are the chloride channels sitting here and why they are important and what's their functional role. Before we even jump into it, the usual question I have to answer is chloride really important for physiology? So I give a simple example of myocardial infarction, which we focus on in our lab. All we did for this study was we hang the heart and did a Langendorf reperfusion assay. And the heart was hanged, we stabilized it, that's the green. Then we either caused global ischemia to this heart for around 30 minutes, or no ischemia, this is our control, followed by reperfusion. And during reperfusion phase, we change the chloride concentration. The normal chloride concentration in our body is around 105 millimoles, which is mostly in the brown color here. And if you go down, we have a clinical data now that your chances of survival after heart attack or any cardiac insult are quite high within first 12 months. But what we did here is this is a rat heart. These are sham. That means there is no ischemia, and these are hearts after ischemia and reperfusion. Red is the healthy tissue, white is the infarcted tissue. I'm going to refer to this again and again in some of my other slides. That's why I just want to make it sure that I convey the message. And when we quantify it, you can clearly see, usually we have around less than 10% infarction if you just hang the heart and do not subject them to ischemia. But the moment you put them on ischemia, you will increase the infarction. This is expected. But look at the low chloride. Even in the sham condition, you really have higher infarction. And on ischemia reperfusion, you do increase this myocardial infarction. Not only that, if you do in vivo assay, so in this case, we reduce chloride. And in this case, we actually used a chloride channel inhibitor, IAA94, which is not very specific. This is a normal problem with all the chloride channels. The drugs are not highly specific, but IA94 at a concentration which we use around 10 to 30 micromoles is highly selective for CLICs, but it does not really act on other chloride channels or other ion channels. So but in this study, what we have done is we ligated the coronary. This is the suture. And then we'll open the suture after 30 minutes and let these animals sit for 24 hours before we sacrifice them. This is again the TTC staining of the heart. What I've shown you here is that if you perfuse these hearts with IA94, you will have higher infarction. This is normalized to area at risk, and this is normalized to left ventricle. Whether you have low chloride or whether you block a chloride channel, you do increase myocardial infarction. That is the take-home message here. Now, the next question is, what about mitochondria? I touched upon mitochondria. So all we did here was we changed the chloride concentration in extra mitochondrial space. This is our isolated mitochondria from 0 all the way up to 110. And we are showing that chloride concentration can affect your mitochondrial calcium capacity. It can also affect your mitochondrial membrane potential. And in fact, this chloride can also affect mitochondrial ROS generation. And if you block these chloride channels with IA94, you do reduce the calcium capacity of mitochondria. And cyclosporin A, which is known to protect these mitochondria or increase the calcium capacity, is not really doing anything if you have IA94. Again, pharmacology or independent chloride studies have shown, we have shown now that chloride is important at organ as well as organelle level. But the question is, what are the channels we are targeting? So IAA94 was originally used by Alak Vethi's group from Columbia University, and they published in a couple of papers, which are highlighted here, that with affinity purification, they isolated a protein at the weight of 64. They called it P64, and they showed that it's localized to the intracellular organelles. So these were the two key papers in the field in 1989 and 92. Moving forward, these proteins were renamed as CLICs, chloride intracellular channels, depending on their function. And I will just summarize all that work in few of these highlighted points. There are 6 mammalian homologs known so far, CLIC 1 to 6. In my previous presentation in person, I presented on CLIC 6. So those are the 6 members. In flies, we have 1. C. elegans, we have a couple. And in plants, we have 4 of these homologs. Even in bacteria, we have characterized a CLIC protein, which is called stringent starvation protein. These proteins are quite unique. They are soluble as well as membrane. So most of the protein is present in the cytosol, but then there is a signal which happens, which we are still trying to figure out. It just goes to the intracellular membranes and form a channel. They are structurally homologous to GST, glutathione S-transferase, omega fold. And these proteins can be blocked by IA94. Another unique feature, they have a single transmembrane domain, makes it very interesting to study. And our work almost 15 years ago have shown that 4 of these monomers come together to form a functional channel. So it's a tetramer. Functionally, several groups have now shown that CLIC4, for example, is involved in apoptosis, pulmonary hypertension, angiogenesis. And if you remove CLIC1, you will have cell cycle arrest. See, these animals are obese. And almost all CLICs are implicated in cancer. But if you go and ask anyone who's working with CLICs, what are the mechanisms involved, where are these proteins specifically present, that information is not present. And I think that's why these proteins are still listed as dark ion channels. So the soluble structure is known. That's the soluble structure of a CLIC4. It's very similar to GST. You can actually overlay it, and every single domain overlays on GST. And the membrane structure is, this is what we know so far. So we are very open to collaborations. If anyone can help us with any of the CLICs to solve their membrane structures with cryo-EM or anything, we are always looking forward to collaborations. We are actually pursuing these collaborations right now on different proteins, but hopefully in near future, I will present a real structure here rather than just putting these cartoons. But they do form channels. If I do planar bilayer studies, I can record beautiful currents, and they are redox regulated. I can block them with IA94, whether it's from E. coli or it's a recombinant protein from human origin. So we have these channel-like function. We have these proteins sitting in intracellular organelles, but that's where we are pursuing our projects right now. So in E. coli, as I was mentioning, we characterize them that they are channels. And if you label these proteins, they are sitting in the membrane of these E. coli. Now coming to mitochondria, if you take mitochondria and patch them, you can actually record chloride currents. You patch outer membrane or you patch the inner membrane, you can always get these beautiful currents. And they are actually highly sensitive to IA94. Remember, this is a compound which was originally used to purify these proteins. So yes, click proteins are present in mitochondrial membranes. But again, out of 6, which one is present and what is their physiological role? That was the original question. So just summarize, as my original title was that I will be illuminating some of these clicks. All the clicks are intracellular except click 1, which can spill over to the plasma membrane if you overexpress it, but other proteins are mostly present in intracellular organelles, as we have highlighted here. This is from our review. So the question was, are these clicks present in heart? And if yes, which ones are those? So what my postdoc did, she started her duration or tenure in our lab by just doing a quantitative PCR. This was the very first experiment she did, and she found that out of 6 clicks, 1, 4, 5 are the highly abundant. Click 2 is absent in mice, which is quite interesting again. So we pursued this 1, 4, 5. And when we did Western blot, we found 1, 4, and 5 are present in the heart. And then we went on to localize these proteins. So these are isolated adult cardiomyocytes, and on the right-hand side are our neonatal cardiomyocytes. In both preparations, what I'm showing you is that red is mitochondria, blue is nucleus, and green are the specific clicks, whether it's click 1, 4, or 5. 4 and 5, they localize to mitochondria. You can see 5 is really beautiful. You can use it as a mitochondrial marker these days. And even in adult cardiomyocyte, it's sitting in there in mitochondria. Flies, they have only 1 click. So we took the cardiac tube and labeled them with a mitochondrial marker. This is ATP synthase and click antibody. Again, even in fly heart, clicks, the Drosophila click ends up in cardiac mitochondria, cardiomyocyte mitochondria. This is a quantification of this co-localization. Long story short, what we have shown you is that click at 4 and 5 are associated with mitochondria. So what we did next is we purified these mitochondria further by Perkol gradient method. And as you can see in our preparation, we don't really have plasma membrane, nucleus, ER, or Golgi in the M3 fraction. But we do have mitochondria in this fraction, which is indicated by VDAC or COX2. And in this preparation, we do find 4 and 5. 4 is not that abundant, but 5 is highly abundant. And these are our antibodies. So we always characterize antibodies using knockouts. So these are highly specific antibodies. What else we did, we took these mitochondria, we loaded them with MitoTracker, and we spread them on a cover slip. These are lamine-coated cover slips. And red is your mitochondria. Green are our antibodies for clicks. And again, you can see click 5 highly localizing with mitochondria. Click 4 also localizes with mitochondria loaded with MitoTracker. Click 1 does not really co-localize that much. So this is our independent assay, because we did get criticized that maybe your protein is sitting in some other organelle which is next to the mitochondria. So this is we are quite confident that these are associated with mitochondrial membranes. So while doing this, what Devasena observed that click 4 sometimes disappear from M3, but it always comes up with mitochondria. So she was curious because at that point, she was purifying mitochondrial associated membranes. And what she did, she just loaded mitochondria and MAMs together. And in this preparation, you can see GRP78 is here in MAM, but not in mitochondria. And click 4, the lower band here, ends up in mitochondrial associated membrane. So this is the first evidence to show that click 4 ends up in mitochondrial associated membrane, but not that much in mitochondria. Reviewers did ask us to do this experiment with several different MAM markers, and we did this. And whatever marker we use, we do see click 4 associated with mitochondrial membranes. So mitochondrial associated membrane is a structure is a membrane which is in between mitochondria and ER. So it's basically tethers ER membrane to mitochondria. Is it a compartment? It's not quite clear because in EMs, you just see a dark region there. So what we did, we isolated these preparations and we reconstituted them in planar bilayer. And we do see current. I agree that we do get criticized for this, that planar bilayers can have some sort of artifacts, but I'm quite confident this is a chloride current because we use NMDG-Cl in our recording solutions, and these activities are highly sensitive to I-94. So we do have some CLIC4-like currents in mitochondrial-associated membranes. Now, next question we wanted to know was, how is this CLIC4 getting to MEMS, right? What is the region involved? So what Devasena did, she put a flag tag at the end terminus of CLIC4, and then she transfected hex cells. And you can see that it does associated with mitochondria. Then she removed half of the C terminus. There is a cysteine, which is well conserved in all the cliques. It's a 189. Again, it ends up in mitochondria. Then she removed all of the C terminus, just kept transmembrane domain with a small, around 35 amino acids in the N terminus. And again, it ends up in mitochondrial associated membranes. So this is our evidence that CLIC4, actually, the transmembrane domain is sufficient for this protein to go near mitochondrial associated membranes or in mitochondrial associated membranes. So mitochondrial associated membranes are very well characterized for calcium transfer from ER to mitochondria. So we were also trying to figure out whether that's true for our CLIC4 as well, whether CLIC4 has any role in this function. So what we did with the collaboration, Dimitri's lab here at OSU, we transduced these neonatal cardiomyocytes with GCPR. This is a calcium sensor, and we measured the calcium release with hepsicargin, and it can take around 400 seconds for these neonatal cardiomyocytes. But for CLIC4 knockouts, you can clearly see the tau has decreased from 400 to all the way to 72 seconds. So the leak is really, really fast, or calcium release is very fast. Where is it going? So we also checked with mitochondria calcium. This is a calcium sensor. And again, you can see that CLIC4 has a higher amount of mitochondrial calcium. We independently corroborated this by our calcium retention capacity. Those who are not aware of it, this is the assay where we isolate mitochondria. We keep on adding calcium. And then at one point, which is the last point here, so as soon as you add calcium, it goes down every single time. But at this point, it will not go down. In fact, it will stay up and go up. That's where we say the MPTP has opened. What we realized is that CLIC4 has reduced calcium capacity as compared to wild type or CLIC1, which is just a control for clicks. And this corroborates the data here that there is extra calcium release or more calcium release happening in knockouts, which ends up in mitochondria. And that's why we have less calcium capacity in mitochondria isolated from knockouts. Now, calcium plays an important role in cardiomyocytes' health, whether it's beating or cell death or myocardial infarction. And I started my story with myocardial infarction. So we actually did quite comprehensive studies using different types of models to see whether CLIC4 has any role in myocardial infarction. And remember, IA94 was increasing myocardial infarction as well. So what we did was, sorry, I'll go to that. We ligated these hearts and did ischemia reperfusion, just as shown earlier for rats. So this is ligation for around 60 minutes and reperfusion for 24 hours. And we also did cardiac functional analysis by doing echocardiography. As you can see here, after ischemia reperfusion, we see more myocardial infarction in CLIC4 knockouts as compared to wild type. And but area at risk was the same for both animals. And the ejection fraction goes down and fractional shortening goes down. This is in vivo ischemia reperfusion. We also did ex vivo ischemia reperfusion. And in this case also, we do see higher myocardial infarction. Remember, the white part is the infarcted part, and CLIC4 has more myocardial infarction after ischemia reperfusion. We also did a long-term ligation for the heart. And again, what we have noticed is that after 28 days, we see higher myocardial infarction in CLIC4 knockout. So 3 different models. One is permanent ligation, another one is ischemia reperfusion, and one is ex vivo Langendorf. In all 3 conditions, we do see that absence of CLIC4 is detrimental. Now, what about the calcium capacity? So when we isolate mitochondria from these hearts after Langendorf, we do see that calcium capacity is significantly reduced as compared to wild type after ischemia reperfusion, indicating that It's actually calcium which is pushing these hearts towards higher myocardial infarction or cells towards the cell death. So there was a question by reviewers that is MCU involved or what is going on? Why your cells are showing higher calcium? So we did a lot of studies on this aspect. We tried to check whether MCU expression has changed. No, MCU is the same. Cyclophilin D is the same. Almost all other calcium channels and transporters were exactly the same between wild type and knockout. So there was no change in the expression. All we were seeing was the change in the function. So in fact, cyclosporin A was not able to rescue this as compared to wild type. So what Devasena did, she tried to see whether CLIC4 has any role with ryanodine receptor. So she did immunoprecipitation, and in this assay, she did find that ryanodine receptors, they interact with CLIC4, which is here, this band up there. It's absent in the CLIC4 knockout, obviously. So one of the possible mechanisms which we think is going on here is that CLIC4 physically interact with the ryanodine receptor and controls the calcium release, and that's how these hearts are protected, or hearts are not pushed towards myocardial infarction or cell death if CLIC4 is present. Its absence will increase the calcium release. Now, what about the cell death? I'm using this term again and again, but I've not really shown any data on cell death. So this is a tunnel assay on neonatal cardiomyocytes. We did exactly the same hypoxia reoxygenation, 6 hours of hypoxia, 12 hours of reoxygenation. And then we did tunnel on these neonatal cardiomyocytes. As you can see in wild type, we do have some cell death as expected, but knockouts, we have a lot more cell death, which is quantified here. So yes, absence of CLIC4 does push cells towards apoptosis on hypoxia or ischemia. So what about membrane potential and ROS? We did all these assays, and again, after hypoxia and reoxygenation, what we are observing is that absence of CLIC4 does depolarize CLIC4 absence, mitochondria lacking CLIC4, and it also increases the mitochondrial reactive oxygen species. So your calcium is messed up, your membrane potential is not good, and then your mitochondria are producing more ROS. So all of these will point to more cell death in absence of CLIC4 on stress. What about expression of CLIC4 after ischemia reperfusion? So we did this assay where we took these hearts, we did immunohistochemistry, and what we see is that there is an increase of CLIC4 expression at protein level after ischemia reperfusion in mice. And we also use human hearts, and even in human heart, these are the failing hearts. We do see increase in expression of CLIC4. It seems that hearts are somehow trying to increase the expression of CLIC4 to protect themselves. That's what is our hypothesis right now. We can easily test this by increasing the expression or by activating these channels. Right now, we don't have good specific activators, but we are actively trying to pursue that, and hopefully we will come up with an answer whether activating CLIC4 will be protected. But to summarize what I've shown you, that there is a novel chloride channel. It's a chloride intracellular channel sitting in mitochondrial associated membranes. Its absence will increase leakiness of calcium through these ryanodine receptors, and that calcium increase will depolarize mitochondria, will increase ROS, activate the mitochondrial permeability transient 4, which will push cells towards cell death and increase myocardial infarction. So with that, what I would like to thank is all my trainees in the lab. Devasena, I mentioned, she's going to start her lab. Shubha did all the fly work. She's now a faculty in Ohio Northern. Ankur did lots of echo analysis. He's now a faculty in Cornell. Veronica is doing all the electrophysiology. Sridhar is a graduate student who did lots of imaging these days. Dimitri and his lab, his postdoc, Shana did calcium studies for us. Adam helped us with the mitoplast. Earlier, Wali and Array did all the ischemia reperfusion. Andrew Cohort was instrumental in doing all the echoes in our initial days, and all the funding from American Heart Association for me, my lab, as well as NIH, in fact, RO3 as well. And in the end, I would like to really highlight our conference again. If you are interested in learning about all these organelles channels and transporters, please contact me. We still have few spots left for poster presentations and oral presentations. And believe me, it's in Barcelona. It's a beautiful place. With that, I will end and we'll take any questions. Thank you.